Hi everyone! Today we'll be talking about the retroviruses. So the retroviruses are positively single-stranded RNA virus family, but unlike the other positively single-stranded RNA viruses, which belong to Baltimore Class 4, and its positive single-stranded RNA can be directly translated, can be directly used to make peptides because it's the same as mRNA. This Retrovirus family belongs to Baltimore Class 6, which means that the single-stranded RNA has to get made into double-stranded DNA, and then this double-stranded DNA has to get integrated into the host genome before the essential viral proteins are produced. And examples of the retroviruses include HTLV, or human T cell or human T lymphocyte virus, or the human immunodeficiency virus, and also the koala retrovirus, which causes leukemia, neoplasia, chlamydiosis in a lot of koalas, especially in the southern region. And this is the structure of a retrovirus in short, and you can clearly see that you have two copies of the single-stranded RNA in its genome. It has a 5' cap for both and a poly A tail. They are loosely bound together near the 5' end, and there is also a binding site for tRNA. And essentially, it moves into the cytoplasm where DNTPs or nucleotide triphosphates are and the bases are, and then the reverse transcriptase makes a DNA molecule from the single-stranded RNA and then makes a double-stranded DNA using the single-stranded DNA. So in the end, you get this double-stranded DNA, which then can be carried by the integrase enzyme through nuclear pores into the nucleus, where it integrates into the host genome. However, important features to note are that the reverse transcriptase tends to make lots of errors and introduce mutations for once in every 10,000 bases. And they can switch between the two different copies of single-stranded RNA. So recombination is definitely a possibility. Additionally, within the nucleus, the integrase doesn't just integrate this proviral genome into any host genome. It is usually in undifferentiated cells, and it is also usually done in transcriptionally active genomes. Except for HIV, because HIV can do this even in terminally differentiated CD4 plus T cells. Now, once in the nucleus it gets integrated, you can get the transcription of this proviral mRNA by host RNA polymerase II. And additionally, this can be an endogenous kind of transcription, so it is passed on. And also you can get the splicing of this genome by host factors as well. And then within the cytoplasm, you can get this mRNA can then move into the cytoplasm where the ribosome and tRNAs are, and then these important proteins can then be made and assembled and packaged in the endoplasm. They can be made in the endoplasmic reticulum and then packaged into the Golgi, into vesicles where they travel and then are assembled at the plasma membrane when it matures, buds, and exits from the cell. And this included in this packaging are also unspliced mRNA. Okay, thank you so much for listening. See you next time.